Hi, alright, we're going to talk about carbohydrate. Okay, so as we all know, carbohydrate divided into three types. We have monosaccharide, the simplest one, disaccharide, when two monosaccharide combine, and then polysaccharide. So there are a few examples, but I'm just going to talk about glucose. Yes, glucose, fructose, and so on, but I'm just going to talk about glucose today. Disaccharide is a combination of two monosaccharide or two monomer to produce maltose, and there are a few more. And last but not least, there is polysaccharide, which are cellulose in the cell wall of a plant. It's in the cell wall of a plant to give the structural support. And then we have starch from alpha glucose, and then we have glycogen, also made up of alpha glucose. Okay, starch is a storage molecule in plants. Glycogen is a storage structure in animal. Alright, so now there are two types of glucose, alpha glucose and beta glucose. These are the structure and how you want to differentiate between those two by looking at carbon number 1, okay, based on the hydroxyl group. So if you look carefully, you can see that hydroxy group at carbon number 1 for alpha glucose is at the bottom, okay, dekat bawah. So tengok ni, saya akan highlight. Okay, while for beta glucose, ingat B, balloon, the balloon will release upwards, so the OH group is on top of carbon number 1. Right? So these are below the ring, these are on top, above the ring. Okay, these two are two difference between alpha glucose and beta glucose. Okey, bagi siapa yang tak tahu, carbon tu dia ada kat sini. Ini carbon number 1. Dan kita akan kira ikut arah ikut arah jam. Okey, carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, carbon number 6. Okey, ada 6 carbon. And student selalu lupa untuk tulis atom hidrogen dekat bahagian dalam ni. Okey, selalu tertinggal. Alright, so yang lain-lain sama, carbon number 2, carbon number 3, carbon number 4, 5, 6 semua sama. Oxygen, everything is the same, it's just OH dekat carbon number 1, satu kat bawah, satu kat atas. Alright, so now you have to be able to recognize and draw the formation how maltose form. So kita tahu glucose tambah glucose akan jadi maltose. Monosaccharide plus monosaccharide akan jadi disaccharide. So if you look carefully, kita just lukis balik alpha glucose. Okay, for your information, maltose is made up of only alpha glucose. Okay, alpha glucose. So awak lukis balik alpha glucose di mana OH tu berada dekat OH tu berada dekat bawah. Sorry ya, lukisan ni silap. Okay. OH tu berada di bawah carbon number 1, dan kita letak tanda plus, alpha glucose plus alpha glucose. And then you have to circle the removal of water. Sebab proses ni adalah condensation. Condensation menghasilkan air. Produk dia adalah water. So awak perlu bulatkan. Okay. And then buat anak panah. Write down the process condensation. And then there are two products. Okay. The two alpha glucose now joining up, combine jadi maltose. Dan dihubungkan dengan bond, okay, sebabkan dah hilang tadi 2H dan 1O. So kita cuma perlu combine line garis daripada carbon number 4 kat sini, okay, dengan carbon number 1 kat sini. Carbon number 1 ni tinggal oksigen, carbon number 4 kat sini cuma tinggal bond saja, tak ada atom. So we just connecting the two lines, and oksigen yang masih tinggal tu kita tulis balik kat sini. So bond ni, the both bond kita panggil alpha sebab melibatkan alpha glucose. Dash 1, 4. 1 tu merujuk pada carbon number 1 dekat sini, dan carbon number 4 kat sini yang membuat bond. 1, comma for glycosidic bond. The name of the bond is glycosidic, and then plus water. Water ni datang dari mana? Dari yang kita keluarkan tadi. And these all will give you marks. Okay, the whole drawing must be correct to get 1 mark. And then nama proses kena tulis for marks, and then label the bonds. And then you also have to label each of the structure that you draw above. For example, atas ni alpha glucose, so tulis kat bawah dia alpha glucose. Atas ni tanda plus, tulis tanda plus. Okay, same goes to everything that you draw above. Okay, mesti align, mesti selari. Tak boleh lukis maltose kat sini, label maltose kat ujung. Okay, mesti di bawah, betul-betul di bawah. Right, so this is a perfect drawing. Regardless, I'm not using a ruler. You are required to use a ruler in the exam supaya gambar awak tu jelas dan kemas. Okay, and then bila benda tu boleh combine, benda tu juga boleh breakdown. So awak cuma perlu ubah produk yang sebelah kanan pergi sebelah kiri. Okay, maltose dan water ni kita ubah tempat. Okay, duduk sebelah kiri, exactly the same. Okay, the same bond. Okay, label juga. Okay, alpha 1,4 glycosidic bond. And then what happened? We add the water. Hydrolysis prosesnya hydrolysis. Kita hydro mean water, lysis mean pecah. Kita pecahkan air tu, air yang dipecahkan tu dimasukkan dalam structure ni. Untuk pecahkan structure ni menjadi 2 separate molecule. So, we going to draw the same thing on top. Yang sebelum ni jadi reactant, sekarang dia jadi produk. So, bila dah pecahkan H2O ni, kita masukkan balik dekat carbon number 1, carbon number 4. Masukkan O, masukkan H, masukkan OH. So, dia akan hasilkan produk yang sama macam awal tadi. This is the breakdown. Okay, this is the breakdown of maltose. Sama, kena label water plus maltose. Okay, and then you will get alpha glucose plus alpha glucose. Alright, that's it for carbohydrate. Thank you.